Hello guys, welcome to our channel. In this video we are talking about gram-positive bacteria, Staphylococcus aureus. Staphylococcus belongs to the family Micrococci. This family consists of gram-positive cocci. Their genus is Staphylococcus. This term is derived from Greek word Staphyl means bunch of grapes, while cocos means berry. It is a combination. Staphylococcus is a combination of two different words, and meaning bacteria occurring in grape-like clusters or berry-like shapes. Staphylococcus may be coccus, diplococci, tetrads, and clusters. As you can see, like in this picture, some organisms are single, diplococci, tetrads, and clusters in form. Now history. First scientist is Robert Koch. He first see the Staphylococcus in pus specimen. Louis Pasteur first to cultivate in liquid medium, and Alexander Ongston named the bacteria as Staphylococcus. They are the three main scientists in the history of microbiology. Now the classification. Based on the pigment production, Staphylococcus are divided into Staphylococcus aureus. They show golden yellow pigmented colonies, while Staphylococcus albus, they gives white colonies, and Staphylococcus citrus, they gives lemon yellow colonies. It is a picture of three differentiated colonies of these organisms. Now, based on pathogenicity, pathogenic organisms include Staphylococcus aureus, while non-pathogenic includes Staphylococcus epidermidis, Staphylococcus albus, Staphylococcus citrus, etc. Now, what is pathogenicity? Pathogenicity means the property of microorganisms to cause disease. Now, classification based on coagulase production. Coagulation, it is a protein enzyme produced by several microorganisms that enables the conversion of fibrinogen into fibrin. This coagulase test is a biochemical test used in the laboratory to distinguish between different species of Staphylococcus. Coagulase positive organisms include Staphylococcus aureus, while negative includes Staphylococcus epidermidis and Staphylococcus saprophyticus. As you can see in this picture, coagulase positive shows the coagulation and aggregation like sample in a test tube, while negative have the clear sample. Now, Staphylococcus aureus the natural habitat of Staphylococcus aureus is nostrils and skin. Their morphology includes they are gram-positive cocci, occur characteristically in groups, also singly and in pairs. They appear irregular grape-like clusters. They are non-motile, non-sporing, and few strains are capsulated. As you can see, these are the morphology of microorganism. Now, culture. When we talk about the culture of Staphylococcus aureus, they are aerobes and facultative anaerobes. On nutrient agar, they appear as golden yellow and opaque colonies with smooth surface. In this picture, you can see the opaque colonies or golden yellow colonies. On blood agar, they appear as golden yellow colonies by a clear zone of hemolysis. They gives beta hemolysis. As you can see the zones of inhibition. On Meconic agar, they appear smaller colonies than as compared to the nutrient agar, and they are pink colored colonies due to lactose fermenter. And on mannitol, salt agar, MSA agar, they appear as yellow colonies as they ferment mannitol. MSA is a useful media for recovering Staph aureus from fecal specimen when investigating from food poisoning. Now biochemical test. Catalase test gives positive results. Oxidase test gives negative results. While Staph aureus, these organisms ferment glucose, lactose, maltose, sucrose, and mannitol with the production of acid, but no gas production. Mannitol fermentation helps in diagnosis process. These sugar molecules produce acid, not gas. Now, virulence factor. The cell wall associated structures includes: number 1 is capsule. Through this structure, the organism adhere to a host cell. They attach to the host cell through this structure. And peptidoglycan, they inhibit the inflammatory responses. And next is extracellular toxins. They include enterotoxins. Enterotoxins act on ANS to cause diseases, while TSST, which is toxic shock syndrome toxin, produce fever, skin rashes, diarrhea, conjunctivitis, and ETH to shock. Next is pathogenesis. Staphylococcus aureus adhere to damaged skin, mucosa, or tissue surface, or any other wound or injury. At these sites, they evade the defense mechanism of the host. They enter in the host mechanism and destroy the defense mechanism, colonize, and cause the tissue damage and cause disease. They produce Staphylococcus aureus produce diseases by different types, by different methods, by multiplying in their tissues, by liberating toxins, and by stimulating inflammations. These organisms enter in the host cells and multiply in their tissues, liberating toxins and stimulate inflammation, so the disease can cause the can become severe. Now, clinical syndromes. These are the infections which can caused by Staphylococcus aureus, which contains some upper layers of cutaneous infections, deep infections, and other infections. Number 1 is cutaneous infection, which is folliculitis. It is the inflammation of hair follicle. A small red bump or pimple like develop at infection site of hair follicle, as you can see in this picture, and then they cause diseases, severe disease. Next is furuncle, which is also called boils. It is a deep-seated infection originated from folliculitis. If the infection of folliculitis extends from hair follicle to neighbor tissues, it causes redness, swelling, and severe pain. Commonly found on neck, armpits, and groin regions. It is the severity of folliculitis infection. Next is carbuncle. Carbuncle is an aggregation of infected furuncles. Carbuncles may form large abscesses. It is a large area of redness, swelling, or pain punctuated by several sites of drainage pus. Next is impetigo. It is a very superficial skin infection, most commonly occur in children, usually produce blisters. or sores on face, neck, hands, or diaper area. It is characterized by watery bristles which become pustules and then honey colored crust. Next is osteomyelitis, which is a deep infection. It is the inflammation of bone. Bacteria, which is the Staphylococcus aureus, can get to the bone through bloodstream or any other injury or any wound. Their clinical features includes pain, swelling, defective healing, etc. And their diagnosis includes X-ray, MRI, and bone aspirates. Here is the picture of this infection. Next is periostitis. It is the disease in which inflammation of periosteum occurs. Their clinical features include fever, localized pain, leukocytosis, and their diagnosis includes needle aspiration of subperiosteal fluid. Next is endocarditis. It is the inflammation of the inner layer of heart, which is the endocardium. This disease occurs when the bacteria enters bloodstream, travel to the heart through blood circulation and lodge on abnormal heart valves or damage heart tissues. As you can see the difference between normal aortic valve and abnormal. Next is exfoliative disease. Exfoliate means scaling of tissues in layers, and it is also called staphylococcal scalded syndrome. Epidermal toxin produced by Staphylococcus aureus at skin and is carried by bloodstream to the epidermis, which is the upper layer, where it caused a split in a cellular layer. This toxin separates outer layer of epidermis from the underlying tissues, and as a result the scaling of layer or scaling of skin appears on a body. Next is toxic shock syndrome. Toxic shock syndrome is caused when TSST, toxic shock syndrome toxins released by Staphylococcus aureus enters bloodstream. It is a multi-system illness characterized by high fever, headache, vomiting, diarrhea, kidney failure, skin rashes, conjunctival redness, etc. Now, staphylococcal food poisoning. It is caused when consuming food in which Staphylococcus aureus has multiplied and formed endotoxins. Symptoms are nausea, vomiting, severe abdominal cramp, sweating, headache, etc. So, these are the mode of transmission through which this bacteria transfer from one person to another. The next is prevention. Always wash your hands, keep wounds covered, reduce tampon risk, avoid sharing personal items, and cook and store food properly. Now, treatment and drugs. First is antibiotic therapy, in which vancomycin is commonly used. wound drainage, device removal, and removal of dead tissues. Thank you so much. Thank you.